collection of biomolecular interactions from scientific publications, and as you can see, this is a joint work with many people. So the names highlighted in the color of Yerevane logo are from Yerevane, and the names colored by the color of big logo are from bioinformaticians, and Andrei Fetsky is from Chicago University, and Hrant Khachatryan is leading the whole project from University of Southern California. So the problem we're trying to solve is the following. We have millions of papers written by biologists published in journals, and these papers contain important information for biologists, but no one can read all of this. So what we eventually want is to extract the knowledge from this paper into graphs, like structures like this, from which we can already work using computer-based techniques, algorithms. In particular, the problem we're trying to solve in this project is to extract binding interactions between biological molecules from sentences. So imagine we have a sentence like this, and we want to understand which words in this sentence correspond to biological entities, like proteins, chemicals, or other things, and then figure out what are the relations between these entities. So our input is text, in this case it's just a sentence, and the output is a graph like this. So if you ask a machine learning person to solve any problem, machine learning people want dataset. So, and by dataset we mean not only the texts, because if you ask biologists how much data you have, they will say like we have millions of papers, but it's not useful for machine learning. So it's very hard to get useful information from these millions of pages. You need these answers, basically. You need to give what you expect from the machine learning algorithm to get from this text. So you need to give some examples so that the machine learning algorithm can learn. And that is why we asked our bioinformatician friends to annotate sentences, basically take sentences from papers and then somehow annotate structures like this so that we can teach a machine learning algorithm to do the same on scale. So in short, this slide shows the summary of what happened. So we developed detailed guidelines for annotations, then annotated more than 2,000 sentences based on these guidelines. Then we prepared several baselines, which are just some algorithms which work on this data and produce some result. We wrote a paper and sent to BioNLP workshop, which is the main workshop on biological language processing, and got accepted, so presented there in a month. We have also released these annotations publicly, and I think next week we will also launch a competition on the CodaHub website, where everyone will be able to use this annotated data to train new models and compare with our baselines. So next I'll try to explain why this first part, which is the detailed guidelines for annotations, designing of these guidelines was very hard and important. So it turns out that you give the same sentence to 5 different biologists and ask to annotate binding interactions, they will give you 5 different graphs. So then you have to ask them to kind of come to a consensus, and this is where you end up with a guideline so that everyone should follow. And ideally, in ideal world, you design the guideline and then use this guideline to annotate everything. But in practice, it almost never works. So you design some guideline, you start to annotate, and then after 100 sentences, you realize that there are more problems that are not kind of expected and were not covered by the guideline. Then you go and revisit the guideline and revisit the annotations, and this happens all over again several times. So that's what happened in this case, and usually it's very hard to avoid this. I'll show you a couple of details how the sentences were annotated. So in the end, we ended up with 33 entity types, like proteins, protein families, chemicals, genes, and so on. We allowed overlapping entities. For example, if you have a sentence like this, NFY binds the HSP70 promoter in vivo, here HSP70 is one entity and HSP70 promoter is another entity. So entities can basically be nested in the sentence. And our dataset is one of the rare ones in all datasets where this kind of annotations are allowed. Also, there are sometimes the sentences contain synonyms or abbreviations. So you have different words which look like different entities, but they actually correspond to the same thing. So ideally, in the graph, they should be represented by the same node. For example, in this sentence, I would never realize that PR actually refers to progesterone receptor. It's not that written next to it or written in parentheses. So that's where you need biology expertise. So this is a list of the top 10 entity types. So I asked Lily to write descriptions on how do you decide what objects, what entities are marked as protein in our sentence. So it seems obvious, but then you see different people fighting on how to annotate each entity, and then you realize that this kind of description is important to fix so that everyone will follow the same rules. Then we asked to do the so-called grounding, which means for every entity, we want to map it to some database. So there are databases of proteins, protein families, and we want to map all the words in these sentences in the dataset to those identifiers in those datasets. So we managed to do grounding for proteins, protein families, genes, and chemicals from these databases. And it turned out that this is an extremely time-consuming process. It's usually very hard to find the name in the database, and the search engines in the database websites are so bad that it's better to use Wikipedia or Google to find these names in those websites than their own search engines. And even after all this process, we didn't manage to ground all entities, even like all proteins. For interactions, so we ended up with 3 labels. So when we have an information about binding interaction in the sentence, it turns out that they could be in at least 3 states. So if the sentence says that these entities bind to these other entities, it's very easy. It's a positive interaction. Sometimes the sentence says the opposite. For example, here it says no appreciable binding of this could be detected. In this case, we call it negative. Sometimes the sentence is inconclusive. For example, here it says additional experiments were performed to clarify whether things could in fact bind to human GG1. So it says it's not clear from this sentence what is the outcome of the experiment. So we label these cases as neutral. Even after all of these things, so I presented only a very small part of the guidelines, but even after all of this work, there are many problems with this dataset. The main one that is not listed here is that it's small, so 2,000 sentences is, yeah. But other than that, we were limited only on binding interactions here. And the sentences were chosen from the texts such that the words bind, bound, or binding were present in the sentence. So this basically added some bias to the data set. And because of the process of the annotations, we could not formally measure inter-annotator agreement. So whenever you do an annotation work, it's usually good to have this measure which is calculated the following way. You you give the same sentence to multiple people and then try to compare what is the agreement between two annotations or several annotations. So due to the design of the process that we had for this data set, this was not performed. And this is the reticulogram of our data set. So there is one big cluster. Every node here is an entity, and two nodes are connected if at least some sentence contains binding annotation. There is one large cluster and lots of very small things. Then we developed a couple of baselines, so basic algorithms that work on this data. The most basic ones are so-called co-occurrence baselines. So imagine you have a sentence and you know that these are the entities of the sentence. The easiest thing we can assume is that, let's assume that if two entities appear in the same sentence, then they actually bind. So this will easily get to 100% recall because all pairs were obviously found by this stupid algorithm. And it will have low precision. But this is an interesting baseline and kind of shows what's happening in the data set. We have two versions of this baseline. First one is when we know what are the ground truth entities, so we assume that someone gave us the correct answers. And the second one is when we don't know the entities, we just know the text, and we should run some algorithm, some entity recognizer to extract entities and then take all the pairs and assume that they are binding. So we used this tool called SciSpacey. This is a very recent thing. I think it was published in January this year. And it's an extremely easy to use tool. So you just give a sentence or text to it and it extracts the biological entities. And it's one of the best ones that you can find on the web. So we used SciSpacey to extract entities and do another baseline. The third baseline we used is a system called REACH. It is developed, I think, by the University of Arizona. It's a rule-based system, so it takes a sentence, does some grammatical analysis of the sentence, and then applies some rules to identify the binding interactions. And finally, we used the neural network. So we wanted to design our own neural network for this problem, and we started to design it, and we had a long roadmap on what features we were going to add to this neural network to make it stronger. And last year in November at EMNLP conference, we have seen a paper which had a similar neural network, which had all the features that we wanted to implement and a few more. So we stopped our own development and took their code. Their system is designed for computer science papers, not biology, but it was very easy to adapt. And it's an interesting multitask neural network, so it does three things in parallel. It does entity extraction from raw text. It does coreference resolution between entities, so if two entities belong to the same, two words belong to the same entity. It also has a module to detect these things, and it also does relation extraction. And the good part is that it supports multi-word entities, which are very common in biology, and it also supports overlapping entities that I showed. So here are the results. So in machine learning literature, what you care about is this table. So you take a couple of metrics and run your algorithm and calculate these metrics for the outputs. And you end up with some numbers like this. So the main metric for our data set is F1 score, basically the harmonic mean of precision and recall. For relation extraction, co-occurrence baselines get only 20% if we use SciSpacey entity recognizer, and 35% if we use cold entities. Rich system got 34% F score, and the neural network got 50% F score. But these numbers probably do nothing for biologists, so we decided to do some qualitative analysis. So we found a paper where there was a large figure that was describing the interactions of some entities, and took the description of this figure and tried to see what our neural network can extract from this description. So this is basically the ground rule. This is what we want to be extracted from this paragraph. So the neural network managed to extract only two relations from this entire graph. And the first problem that we identified is that, as you remember, our data set is biased towards sentences where we have words bind, binding, bound. And to see how this was important, it took this paragraph and replaced several words with bind. So this recruit became bind, hydrolysis became bind, and phosphorylase became binds. And after doing these modifications, the neural network found almost all pairs except this one and this one, but it also found lots of other things that didn't exist in the sentence. These are marked by blue. So then we did another modification. So she simplified some of the sentences, removed a couple of things, and replaced complex grammatical things like which in turn was replaced by afterwards, and this entity was repeated. And after these changes, we ended up with something like this, which didn't detect this entity for some reason and this relation, and detected additional relation false positive here between RAF and ERK. And as the system is designed to support nested entities, here you can see that it made an interesting mistake. So it found that GTP and RAF are two separate entities, but it also detected the entire thing together, GTP and RAF, as a single entity. So it shows a binding relation between RAS and GTP, binding relation between RAS and RAF, and RAS and GTP and RAF. So that's all about these examples. How much time do I have? We have developed a web application where you can put your sentence and see how the neural network works. So this is basically what you get if you put a lot of text. You get some graph here. It's not the best user experience you will find, but yeah, it almost works. And then I want to talk about two topics that are less from biology and more from machine learning problems. So one of the things that people usually forget about neural networks is that it's very hard to understand the confidence of the prediction of the neural network. So if you have a neural network that can perfectly recognize dogs and cats, and if you give this network a completely unrelated image, for example, self-organizing mob from your work, it will not say, I don't know. It will say either cat or dog. So there is no easy way to understand that neural network is just saying completely unrelated. And this problem is recently investigated by the community, but usually in image processing and not in language processing. So the question that we are interested here is, can the neural network tell us if it doesn't know the answer? Or to put it more formally, suppose our algorithm got 50% F-score on the entire test set. Can we get a higher score on a carefully chosen half of it? Like, imagine we are allowed to answer only on 50% of the test set, only on half of it. Can we carefully choose the sentences on which we are more confident and get better score? So, there is some research on this, again, mostly on image processing. We took a paper from last last year's ICLR. ICLR is one of the major conferences in machine learning. This paper described a method on how to extract confidence information from the neural network prediction, and we tried to adapt this method for language processing tasks, including relation extraction tasks. And with some tricks, modifications, and things like that, we managed to get a 59% F-score compared to the 50 baseline on the best half of the test set, basically. The other thing that we try to do is to extend the data set with noisy labels. So it's very hard to get ground truth gold standard data, but maybe it's possible to use databases like StringDB and somehow leverage this information in those databases. So here is what we did. We took relations from StringDB that were labeled as experimentally verified. So StringDB has all kinds of relations. It predicts relation between any pair of protein using different methods, but part of it contains like manually curated data. So we took these manually curated pairs of proteins from StringDB and looked for papers in PubMed Central where we have a sentence which contains both entities and contains the verb bind. And we assume that this sentence contains binding interaction between these two entities, which can be wrong in many cases, but that's the price we want to pay. So with this procedure, we found 21,000 sentences. By the way, we expected a lot more than that, but for some reason we found only 21,000, which is 10 times more than our gold standard. So as you can see, this process has many problems. So this noisy data set, not all entity types are covered. So StringDB does not contain all the entity types that we have annotated. Also, for each sentence we have only one pair, basically, and in real life we have like 1.5 pairs on average. And sometimes the same sentence contains too many entities, which means that if there is a word bind, it's very hard to know to which two entities this bind refers to. But nevertheless, we tried to use this data set, and the results were terrible. So if you train on gold data and test on gold data, you get like 50% F-score. If you train on this data, I forgot to say that this method is called distance supervision. Basically, you're trying to supervise with some distant source. So this distance supervision data, when you train on it and evaluate all the gold data, you get the result of 25% F-score. And even if you mix the two, it actually harms the performance. It's not bad. This is actually also a very important detail that in machine learning, sometimes low quality data make things worse. So if you can somehow filter out the bad data, it might even help. So we used a couple of methods to identify what are the worst parts of our distance supervision data, and it actually helped a little bit. So if the combination of gold data and distance supervised data, the full distance supervised data, get this score, if we choose the best 50% according to some metric, this 41 became 47. And if we chose the best 1,400 sentence, which is exactly the number of the gold standard sentences in the training set, we got to 50%, which is basically the same as gold data. So if we carefully choose the best part of the distance supervised part, that data, it doesn't harm the performance. But obviously we wanted to have much higher number here, and this didn't help. couple of words about Yerevane. So, Yerevane is a relatively young lab. So yesterday, Hans told in his talk that bioinformatics group came to the game very late. So Yerevane came even later. And we continue to think about future directions. So now we are going to grow the lab. And our current understanding is that we are going to have two types of projects. So first will be pure machine learning research, which we already started to do with some professors from USA and France. But also we will have a couple of projects about applying machine learning to other sciences. And we are open to interesting ideas in both directions. Thank you.